So, hello everybody. Tonight I will show you how you can find sequences in sequences, and in this tutorial we will basically talk about how we can find DNA sequences in DNA sequences. So if you have a subsequence, or you want to find inside one of the human chromosomes, for instance, locations of a given sequence, you can use this tool here, and you can use basically the methods that I'm explaining to you here. And I hope you find this useful. This will go a bit into theory around the Fourier transform, and basically things that are called the convolution theorem, and how you can use the convolution theorem in order to, as we just said, find DNA sequences in DNA sequences. And I wrote a little program in order to do this. This is also then a tutorial to this little tool here. So what are we talking about? So this is called the Fourier-based sequence in Sequence Finder. And what do you need for in order to use this? You can find a sequence in a FASTA file. So basically you need a FASTA file where you have this sequence inside of, for example, a whole chromosome, and then you need a second FASTA file with the little subsequence that you actually want to search inside this whole chromosome. Yeah? This is fast and accurate, so it's very, it runs in under 1 minute at least on a decent machine. If you search the human genome chromosome 1, it's accurate, so this means if the target sequence, if the sequence that you're looking for is found within, for example, a chromosome 1, you will get all the positions where it is. Yeah? So it's not like BLAST where maybe you get the position. It is really if the sequence is there, you get the position of the sequence. You can also then use a certain value in order to tell, well, how much difference between the sequence and the sequence that I'm searching for can I admit. And we will go into the details during this tutorial. So where do you find the tool? So this is the Zenodo entry. So if you use this tool, I basically would like you to cite this here. Yeah, that would be really nice. And otherwise, well, the tool itself, it's on GitHub, so you can download it over there. Well, just let's head over there. Let's open this in the browser. So the browser should be coming up. And here you basically have the web browser, and on this website, this GitHub site, you can just download the tool if you want to. Yeah. Okay. So far so good. So how does this work? So the basic idea that we are using here is what we call this signal detection. Yeah. So this is used from the theory of signals. So this is also used in radar, in image search, in all kinds of stuff. And here we use it then to find DNA sequences. So how does this work? So imagine you have a huge signal, and you want to find in this huge signal F. Let's say we want to find the small signal here, which is represented by this little stuff here, and then you do something which is called convolution. Yeah? And the idea is that if you convolute the small signal with the large signal at all positions, you will get a third signal, which is of course the convolution between those two signals, and at the where you have peaks basically in this third signal. This is where you expect the other signal to be. So we can quickly go through this. Yeah. So how does this work? So you take this value here, you multiply it with this value, but then you add this value multiplied by this value, and you add this value multiplied by this value. and if I didn't make a calculation error, it basically should be at this value here then, yeah? So if you add these multiples, basically the dot product, the local dot product between these, and then you slide this across, and for each position you get, for this local dot product, you get a value, yeah? And as you can see, well, if we get over here, well, we get 6, so if you do minus 2 by minus 2 is 4, plus 2, and you get 6. And that way, as this is really here the highest value, you basically have perfectly found the position of this subsignal within the original signal, yeah? And here we will then try to use this for our DNA search. Yeah? We continue with this a little bit within theory. So as I just said, this should also allow us to find gene sequences in gene sequences. This is what we are applying this to. But the big deal that we have here is that if you do this, especially if your kernel gets larger, this is very slow. So imagine you have here the whole chromosome 1, which has like billions of entries, yeah, and here you have a huge, let's say a subsequence of 100 basis pairs, nucleotide basis pairs. And you do this, so you have, for each position, you have to calculate a dot product of at least 100 basis pairs then, yeah? So it's 100 times 1 billion is going to explode, yeah? And this is a very slow procedure if you do this that way. And so the nice thing that we have is that basically Fourier comes to help, so you can use a very nice trick, which is called this convolution theorem, where you can use the Fourier transform in order to solve this problem here. Yeah. So what does this convolution theorem basically say? So the convolution theorem says that the Fourier transform, yeah, of a convolution of a signal with another signal is, the Fourier transform of the individual signals, where you just multiply each point of them. Yeah? Here basically you have the proof. Yeah? So I will walk you through this. So the proof is very easy. So you take this equation here, and you just insert with the formulas that you have here. And then you get basically out that this here is the convolution formula. Yeah? So very easy to know that if you move this across here, yeah, and if you write this as integrals, of course here we are in a discrete space, so these integrals basically you can write them as sums, but it's very easy to see that here this xi here, yeah, is basically this running variable. You just run from one point, where you run this up here, yeah, so you move from one point to the next, and then you get this third signal, this convoluted signal here. Yeah, it's a fancy type of writing to what I've just shown you, that you take this local inner product. Yeah? Okay. And so if you have this, so you can say, okay, so I have this here, and I want to see if this really is the convolution, if I take the inverse transform of this. And so I just set this in here, and here I take this inverse transform, and then I come to this, okay? So this is the first step here, yeah? And then a bit of magic happens, so you can run this, you can take here this g hat here, yeah? So this Fourier transform g hat here, and you can say, okay, well, if this is the Fourier transform, I write this with this auxiliary xi here, and I write it just out as this forward transform here, yeah. And if you do this, then you get basically this in here, that you come then to this xi minus xi term here, yeah. So you see this here with this e minus blah blah blah, and you take this out, yeah. And then of course you can just move this around, And then if you do this, so you can push this inside the integral, which is just a constant, and you can basically either move these parameters around between these integrals. This is well known. And then you find that this here again, is this inverse Fourier transform that you have here, and if you put just this term in here, you just come to this x minus xi term. I hope this is basically just inserting and solving this part. We don't want to go into all the details here. That's the way it is. It's very easy to do. If you want to look it up, if you want to have it more slowly, then I've done it here. You go on Steve Brunton, you search convolution theorem on YouTube, and you will find a more in-depth, very clearly explained situation about this. So, well, right now we have all these tools, so let's get practical. So the idea is we use this convolution theorem in order to solve this problem here, and in order to find, let's say, already nucleotide sequences in nucleotide sequences. No, that's what we actually want to do. And so, how do we do this? In Fourier space? So the first stuff that we do is, in order to get this to work with the Fourier transform, you have to invert the canal. So we have to invert the canal. This means that you have to write it down in inverse order. So here we were at 2, 1, 1, so we write it inverted, so we write 1, 1 minus 2, so that we get here the correct positions out. That's the way how it's done. And once we do this, well, we fill up this sequence that we're searching for. In our case, we do it then with zeros. You will see why this is a very practical thing to do here. And as we do this, then the basic idea is, well, you have your signal, you have your inverted canal, you take the Fourier transform of both of them, so you transform our signal f here into Fourier space, and then we transform this signal into Fourier space. Okay, and then as we have everything in Fourier space, so we just have to multiply element-wise. So each element, as we have actually grown this inverted canal, of course in Fourier space these have the same size then, in K space. And the big deal here is that then you have just to element-wise multiply it, and you get the convoluted terms in Fourier space. So this is just when we go back here, this is just this multiplication here. Okay. So we multiply this, and this is of course way more simple than doing the Fourier transform, than doing this convolution as we did it by hand, where actually for each point you blow it up, because you don't have just one multiplication, but in our case here's 3 multiplications. So imagine this is way longer, then we have way much more multiplications, and so here we just multiply once for each position, which is of course way faster, assuming that the Fourier transform is fast, which it is, it is very well implemented. And once we have this, well, we just do the inverse Fourier transform, and we get to our convolution in real space. Okay, nice, all nice to done. So as I said, well, this is fast too, this is fast too, and that's the way to do it. Well, so this is basically how you can solve this problem quickly. Okay, so now but of course we don't have normal signals, but we have DNA sequences. So you see here nucleotide sequences. So you have here nucleotide bases. So you have here bases. So we have here bases in the DNA, and we want to find, let's say, a sequence of basis pairs, and a sequence of basis pairs in a chromosome, that we have somewhere stored, or in some sequence. Okay. So What we do is we do this, what is used very often in machine learning, which is basically called this one-hot encoding, yeah. And so we take the four bases, these four nucleotide bases here, and we say, okay, well, every time I have an A, I put a 1 in here, and then when I don't have one, then I do a 0 here, and then I get 4 signals. So we basically just transform the original sequence into 4 signals. Yeah. Okay. And then we do just what I've told you with all the signals. Yeah. So we have then 4 signals for our initial sequence where we are searching our subsequence in, and we also have 4 sequences where we for this what we call here the kernel, or the sequence that we are searching for. Yeah. So all in all, we have then 8 signals. Yeah. Okay, so what we do, we Fourier transform these 8 signals, then we get all this in Fourier space, and for all of them, of course, corresponding A to A, C to C, G to G, and T to D, we do this pairwise here, this element-wise multiplication, and then we perform this inverse convolution, and we sum them up for all 4 results. Yeah? And the big deal, if you do this that way, is that we can do something like this, and then if you do this exactly in the way that I've described here, yeah, so that you just sum this up afterwards, yeah, then you will find that you get exactly the number of matching basis pairs. Yeah? So if I'm searching for a subsequence which has 3 basis pairs inside, and in this case, well, I count 1, 2, 3, and I get the result. This is a perfect match. Yeah, and if I have an imperfect match, well, then I just get a lower number. Yeah, this is since this is 0 and 1. So if I have the convolution and this is 1 and 1 here, then I just get 1. Yeah? And that's why this summing up basically works perfectly. Yeah. With these 4 sequences and 4 sequences. And so, yeah, as I just told you, if you have a mismatch, what happens then is that you lower this number here, and you just get, for instance, in this case here, 2. Yeah. Okay, I hope this is very clear. This should be very easy to understand. So that's what we do, and this is pretty fast and works perfectly. And so right now I will show you this. So yeah, okay, I will show you this, but just to mention it, so there are no errors, there are no guesses. If you have the sequence there, you get this perfect matching result. Yeah. If the sequence isn't there, you get a lower number, and that's it. Yeah. And there are no guesses, and so if the sequence is there, it's found. There are no heuristics or anything else like that. Yeah, and the tool that I've written, it sorts you a bed file, and then you can use this bed file in order to see your results, for instance, in this human genome browser, or in the IGV, or any of these tools that basically use bed files. Yeah. Okay. Let's show you how this works. Yeah, so as I told you, we have the code here on GitHub, which is here. So we go here on GitHub, we open this website. It's a bit slow. As you know, my computer here is downclocked, just as I told you last time. Why? Okay, because I have this already open here, sorry for that. So here you have the code, and then it tells you here that you need these dependencies. So again, if you are, just as last time, if you are on Ubuntu, you can install this that way. If you're on a Debian-based system, these are the name of the packages, or the meta packages that you need to install in order to compile this, in order to get this running on your machine. Yeah. For those that are not there, what you need is the FFTW3 library, which you can find very easily, yeah, in its development form, of course. So you need the header files of it, and some distributions here distinguish between the threaded version and the non-threaded version. So you need the threaded version, or it's nice to have here the threaded version in order to use this. Yeah, you need the threaded version actually, yeah, in order to compile this. Yeah. So that actually this runs faster if you have a multi-core machine. Yeah. That's how this works. Yeah, and it just uses all the mechanism that I've just been talking to you about. Yeah. Okay. We have this here. So how? Let's get this installed. So of course I have all these dependencies. If you don't have these dependencies, you just copy and paste this into your terminal, you run this, and you get all your dependencies installed. And if you don't have sudo rights, well, then ask your administrator to install you those packages. You can also install probably FFTVW by hand if you want to. Yeah, it's just you need to make sure that you install the threaded version. Yeah. Okay. So far so good. Then you need to compile this using this command. So I will do this here together with you. So what you do is basically you copy this here, you clone this Git repository onto your machine. So I have here a little machine where we can run this, which has enough memory available. So as you can read it here, this tool needs in order to work with chromosome 1, so if you really want to work with the human genome, it's very nice to have a 64 gigabyte machine which has 64 gigabytes of RAM. Yeah? It also works on smaller machines, of course, but the story is that if you have a real 64 gigabyte machine, you're well served, and this doesn't pose any problems if you're looking for subsequence in ranges in ranges like the chromosome 1 from the human genome. Yeah, so that's what we will do here. Okay, so I copy this here and I will just do like git clone this over here to this machine. And I have my little sequence in Sequence Finder over here, yeah? And as I have my sequence in Sequence Finder, you don't see me anymore because of this browser, yeah? But I'm back, don't worry. So if you have this sequence in Sequence Finder here, we have to compile it. And in order to compile it, you just copy-paste this command here, which compiles and links it to the FFTV3 library. If you are on an ARM machine, you can use this of course too, but this march native, well, just put in here mtune native, something like this. You can look it up in the GCC manual, or you just skip this part here because this is just more or less optimized as much as it is as much as the FFTVW library is optimized there. So you can skip this out if you're on an ARM machine, or you type in here mtune native, because I guess on different architectures this march and the Intel architecture this march command doesn't exist. Yeah, so I will copy paste this here because I have a nice Intel machine here, and I will just copy paste this into my terminal, and I get this. This is compiled right now. And I have this sequence in sequence tool, yeah, so sequence in sequence, yeah, okay. So right now it tells me what I need here, and here we come to the big deal of this here in order to make this work fast, which is this wisdom file, yeah. So we'll talk about this. So here, first what we need. So we need a sequence to search in, yeah. So I prepared this, of course not in this folder, but just to maybe a little look. So I have here this chromosome 1 folder, yeah. And in this chromosome 1 folder, I have my chromosome 1 FASTA file. I already generated the wisdom file in order to show you how fast this goes. If you don't have a wisdom file on a machine like this, it will take like about, let's say, an hour or something like this. So this is a very nice machine. This is an Intel Core i7 machine. Yeah, if you have a less, let's say, the less performant machine, as it has 46 gigabytes of RAM, supposedly, I guess it will be a pretty performant machine, so at least you let this run one day overnight and then you get your wisdom file. So the important thing is to have this wisdom file. Yeah? And we have the sequence that we're searching for, so I can show you this. So this is my FASTA file here, this is my sequence, and I'm searching for this sequence here in my chromosome 1 FASTA file. Yeah? So I will just show you that this is chromosome 1, so let's do it like, in 30, or okay, you won't see that much because at the beginning they're all unknown residues, but so the basis pairs, yeah. But this is, if I scroll back here in this terminal, this is the chromosome 1 file that we downloaded from the internet. It's pretty easy to find, so you go here and do I still have the URL here because I downloaded this here. So I downloaded this file here, yeah. So if you need the URL, you get it here, and you unzip this with gunzip, and you have your chromosome 1 file. Okay, so that's where you get this file. If you don't have it, if you need it, you can just go over here, and on this golden path you get for each chromosome of the HG, so of the human genome in its 38 version here. These chromosomes that you can download. Yeah, this human genome 38 is basically a reference genome that we have. Yeah. Okay. So I will search in the chromosome 1 for this stuff, and how do I do this? So I got my stuff compiled here in the sequence to sequence finder, and what you do is, if you type your sequence in sequence, so what we need the FASTA file, the sequence to search in, so chromosome 1. Yeah? So we're searching in chromosome 1. So I prepared this here in this folder, chromosome 1, and here we have this CHR1 FASTA file. Yeah? Let's just head back again. What else do we need? Or sorry for that, I will just go here so that I get this here. So I have my chromosome 1 FASTA file as first argument, then the sequence that I'm looking for. So I've just shown it to you. I already also stored this in this chromosome 1 folder, and I have here a sequence, so this little short sequence that I'm looking for in this in the chromosome 1, in the human genome chromosome 1. Then a number of sequences to search in. Yeah, so the idea is, when I wrote this, I had a FASTA file where I've, or if you have a multi-chromosome FASTA file here, you can basically you can here tell which sequence in this FASTA file I use. I'm still searching. Yeah. So in this case, this is 1. There is only chromosome 1 in here in this sequence. If you would have, for example, here the HG 38 genome as a FASTA file in one single FASTA file, you can then basically scroll through this and searching then 3, 4, something like this. Yeah. Okay. Just use one and do it with one sequence and one sequence if this confuses you. It's very, very easy to do that way. Yeah. Okay. The next thing that we need is this wisdom file. I already created a wisdom file. If you don't have a wisdom file, you use one here. Yeah, you will also get results, but the story is for the large sequence here that you have. So for the for the chromosome 1 sequence, for instance, if you have a wisdom file, it goes way faster. So the wisdom file, if you don't have a wisdom file, this tool basically runs all types of sorry, you don't see me here, of chromosome, of Fourier transforms. And as it tries out all different types of Fourier transforms, you get into the problem that you that you try them out all, and it tries you to find the fastest, and the fastest it stores in this wisdom file. Yeah. So what I suggest you to do, if you want to frequently search, for example, the human genome, you let this running once at night, you put up a little sequence, you search them in all the chromosomes which are in H338, and then you can run it fast. So if you don't have it, you type 1 here, as we already have the wisdom file, we type 0 here. Then you type the path to your wisdom file. So in any way you have to write a path here, because if you don't have a wisdom file, it will store it there. That's the way it works. So chromosome 1. Then we use this wisdom file. How did I name this? This is chromosome 1 wisdom. So as I told you, if you have frequently searched, for instance, in the human genome, just use any wisdom file. Just run this once for all chromosomes and store the wisdom files. You have them then. If you have this for the mouse or whatever, you just do this overnight, and then you can use this all the time very quickly. So you need these wisdom files to generate them once. I just did this beforehand. It took around 4 here for chromosome 1 on this machine here, which has 8 cores. It took about an hour to generate this wisdom file, or 1 hour and a half or something like this in order to generate this wisdom file. So I already have it. Okay, so I put it here. The next thing is this cutoff. So cutoff number of sequence changes to accept. So if I accept in my target sequence that I have a single nucleotide, how do you call it? Single nucleotide differences. So if I have a single nucleotide which doesn't confirm, I just put it here. So if I basically search for the sequence and I say, okay, I accept this sequence with 2 nucleotide positions differently, then I put 2 in here. In this case, I don't accept any differences in sequence. I just want to find the sequence as it is. You can also use in your search sequence the letter N if you don't know which amino acid to put there, and then also you have to put here for the letter N, well, you have to sum them just up. So you basically just say where you put the letter N, you just say 1 if you have 1 N there, or if you have 3 N's in here, you put 3 N's there. Put 3 here. So this is the number of how many single nucleotides in your sequence you accept to be changed in order to have a valid position. I hope this is clear if I speak it out that way. I don't accept any changes, I only accept perfect matches. Okay. Then the chromosome specifier. Here basically you can put what you want. The story is it will use this in the output, because the output is a bed file, or is a bed file syntax. And as it is a bed file, you have to know the name of the chromosome here, so that you can then use it in the human genome browser, for instance, on the internet. So let's do this. Let's run this here and let's see what happens. So I have run this here with chromosome 1. Did I miss anything? So number of threads to use. So of course here I have an 8-core machine. This is hyper-threaded. 16 cores. This is connected here remote to this machine. This is hyper-threaded. I have here 16 virtual cores. If you don't know it, you can use this nproc command. You can type something like this, nproc. With this replacement here, and then it puts you the number of processors, of virtual processors that you have on your Unix Linux machine. Yeah. Here I put in 16 and I run this. Yeah. Okay. Perfect. And as I do this, so this should be rather quickly on this machine, as we already have the wisdom file, so it might should be less than a minute. I'm not sure that when I tested this on a 28 core machine. There it was at least less than a minute, but here we have 8 cores. Let's see how fast it goes. Yeah. I mean, we are searching the whole chromosome 1, so you imagine you have billions of basis pairs. Yeah, so let's just check it out. If I take here human genome 38, GHR1 size, let's find this here. But no, that's not what I want. So this is the number of basis pairs. So it's 28. So 250 million basis pairs that you're point by point basically performing. Of course, using this Fourier transform. This comparison here. Yeah. Okay, we're still not finished. I'm a bit worried, but I hope it will finish very soon. Yeah. So this is the way it is. So here we finished. You see, we have our little stuff here, and here it tells you the chromosome 1 positions where we have this sequence that I have here. So just recapitalize some 1 sequence. So where this sequence here exists on chromosome 1. So and right now I know it only exists exactly twice. It doesn't exist more often than twice in this chromosome 1 here. And I know perfectly that it's perfect on these 2 positions and that there are no residue differences. Yeah, if I would have found basis pair differences, if I would have accepted different matches, I could have put here 1. We can try this out. Yeah. For instance, we run another run and we accept, let's say, 2 different pairs. So let's do 3. Maybe we get more out of it. Not sure, but let's see. Yeah. So I basically, when I put up this sequence, I just copy pasted a little string from the original chromosome file in order to make this presentation here. And it runs, and I did not know how long it took, but it was on this little machine here. I mean, on this computer here, it was maybe it was a bit above 1 minute, but if you have a big machine, well, you can get it under 1 minute, and you see it's very fast. And the very interesting story about it is doesn't depend on the sequence length that you're searching for. So any sequence length that you're searching for is basically valid, and it takes exactly the same time. Yeah, that's the magic behind this. This is really the magic of this tool. Yeah, so even if you're searching for very long subsequence, it doesn't take more time than this. Yeah, okay. As I told you, if you don't have the wisdom file, of course it takes a bit longer. So if you don't have a wisdom file, you put 1 here, you create the wisdom file, and then it can take hourly long just to find the perfect Fourier transform. But that's only once you run this. Yeah? So if you install this on a cluster, if you install the sequence in sequence tool, right now we found lots of matching positions. So if I scroll back here in my terminal, we see that we got here a whole table of positions because we accepted 3 different single nucleotide positions. Yeah. So I don't want to copy paste this, so you can redirect this into a bed file, and so something like this, and I will call this here. How will I do this? I'm thinking about that here. I will call this here output bed, and then I have a bed file, because as you see, this here is already a bed file, so this bed file syntax. Okay, and then I have to copy this over to my machine because this is a remote machine, but just for demonstration purposes. So we will go to the human genome, how we call it, to the human genome browser, where we can then visualize these positions. So human genome browser. Okay. Here we have the human genome browser here, and then we get here to where are we? Genome browser, interactively watch genome data perfectly. Okay, I'm in Europe, so I will go to the European side. In America, you will have your own stuff. And I'm here, and I'm looking for the human genome. So that's the way it is. I don't want to find the position, but the storage is here is that we can have here my data, and that I can put in here custom tracks. That's what we will do then. So we'll go here, for instance. You can use this here, and you go and see here I have the human genome chromosome X here. I put you here, but you can say, for example, I want to have chromosome 1. That's where we have been, and then you get an overview of chromosome 1. No, what takes here so long? Analytics, Google is so crazy. As web developers, they put all their stuff in here, and then everything is slow because they want to do the analytics stuff visibly, or I don't know. I hope it enters fast. Okay. Okay. Whatsoever. You see that we are here in chromosome 1, and you have this overview of all the stuff down here, and you can even look here at the it corresponds to different species, and where basically the coding regions, and where there are the genes on this chromosome, and all kinds of stuff. I will just make it a bit bigger and maybe zoom out with my little browser here, because I have a very huge font here, because this very small screen is this very small laptop, but with nice resolution. Okay, so we did this, and we have our bed file here right now. So if I show you the bed file, this bed file, we have all these positions here, and we probably want to visualize them. Sorry for that. And so as I have my output bed file, I have to copy it over here to my machine. Let's see what kind of this is using VPN. This is a machine at my lab, so just have to find out here the IP address, which is here. I clear this here, and as I have the IP address here, I can do copy this over here to my stuff, to my machine, so that I can upload it to the browser. I just need to upload it into the browser. Nothing special here. I will just do this here. That way. I don't remember how I named this. I only have one bed file in there anyway, and let's copy this here to my home folder. Okay, then I have a password, so I won't tell you the password, it doesn't matter. I hope I didn't mistype it. Typical me. Other password, let's try it again. We have our bed file here. That's what we needed, so that we can upload it in the browser. Where did I put my web browser, guys? It's over there. So here's my little web browser. And here I can then say custom tracks, and I have my bed file, and I can say browse home, and I have this output bed file that I generated with the sequence in Sequence Finder, and I can submit this up here, and I can say, and I have it right now. And if I go back to the genome browser, then it should be in there. if we don't have this Google whatever stuff. I don't know, to put this Google Analytics stuff and those stuff in there, and then requests take like forever. So crazy. Internet got crazy these days, to be honest. Yeah. And here, so I have sequence position with 3 mismatches. What is there going on here? So this here is my bed file. I had more matches, no? What happened here? And here basically you see with these little traces here, you see where you find your position. So basically they're all positioned around here visibly. Let's see, where's my did I copy the right bed file? Yeah, I have lots of stuff in here. With 3 mismatches, yeah, because we searched for 3 mismatches. And they're all around this 2,600,000 stuff here. Are we there? Yeah, okay, so they're all here, yeah, so they're all at this position here. Yeah, this is here my custom track. And so this way you can basically visualize where on your human genome you have this. So you can also use the IGV browser if you want to. As you have seen this, I will probably stop here with this little tutorial. You have seen how the sequence in sequence finder basically works. I hope you have fun with it. You try it out. If you're looking for a sequence in your huge genomes or whatever, and mouse data and whatever you have, use this tool. It's very fast for this stuff, as you have seen it, and I hope it's helpful for you. Thanks so much.